Okay, I guess it is 1:15 and some people may still be dropping in, but we'll start with tutorial 2. Is that okay?
Sounds good.
Okay, perfect. So we'll be in this tutorial, we'll be going through the basis profile curve identification to understand the effects of electrical stimulation. I just wanted to mention that if people want to walk through all the materials today, I have links here up on the slide. There's a data set available on OpenNeuro and the code is available on GitHub. If you have the opportunity to download that code, it's a Python package. And if you run the first section, that will set up the environment correctly and allow you to walk through all the steps with us. So before we start with our introduction, I would like to thank first of all Kai Miller and Klaus Müller, who have contributed a lot to the development of the method, and Tal Palatia and Harvey Wang, who have been a major contributor to the to making the materials available in the Jupyter Notebook, and then to our collaborators and team here at Mayo, who've been helping with collecting these type of data. So this is a sort of introductory talk will be given by Kai Miller. As you can see on the slide, he is a neurosurgeon, and he's unable to be here today. So I will be playing his talk from a video. And then we'll turn into the interactive session when it's done.
Dora Hermes, who's here at Mayo as well. Hello, my name is Kai Miller from Mayo Clinic. Today I'd like to tell you about a technique that we developed called basis profile curve identification. I'm going to show you how this technique is implemented using an example that was our principal application, namely single pulse electrical stimulation to examine connectivity with patients that have electrodes implanted in their brain. This is work that I did with Klaus Robert Müller from Technical University of Berlin and Google, and also with Dora Hermes, who's here at Mayo as well. The basic question for the application that I'm going to illustrate today is how we cluster responses that come from electrical stimulation, where an electrical impulse is delivered between a pair of electrodes, and a subsequent response is measured elsewhere in the brain. This technique is commonly referred to as cortico-cortical evoked potential measurement. There are a number of ways that we think that these data are most easily looked at, or at least a number of ways that we break down the problem of how these signals can be assessed. The first is something that we call the divergent paradigm. Namely, we stimulate one area of the brain, and then we measure characteristic responses at other areas in the brain. So if we stimulated here at this site, approximately in Broca's area, we measured somewhere else, let's say from the anterior aspect of the middle frontal gyrus, we might get some characteristic response shape, as you see here in blue. And when we see this at site, what we call site A in this example, we measure a characteristic response, and we assume that that response tells us something about the microstructure of the population of neurons underneath one of our recording electrodes. However, if we go to a different spot, let's say here in the parietal cortex, we might measure a response of completely different shape. The problem that we run into is that even though in this example, the output is approximately the same and arrives in superficial lamina to both areas, the response is different because these two areas have different underlying microstructure in the neuronal population. So that has some ambiguity. The same output type could reflect a completely different type of connectivity, or it could simply reflect the fact that different structures are present in different brain areas, which we've known since the time of Brodmann. And of course, we wouldn't just look at two areas, we would look at many areas in this divergent paradigm. Another way to look at these signals is something that we call the convergent paradigm. Here, an electrical impulse is delivered at one site and we measure in another site, again, in approximately Broca's area. And we measure one particular response shape. So we call this response type 1 here in red. But then we measure again from the same site while stimulating at a different site. If we get two different responses here, we know that the types of interactions between the two brain areas where we've stimulated and the area we're measuring from must be different, because the recording is always made from the same site and reflects the same underlying cortical microcircuitry. So different response shapes measured in this context reflect different types of inputs. And of course, we could do this for many stimulation sites, not just two. When we put together the convergent and divergent paradigms, we find what we call all-to-all paradigm. Namely, we stimulate everywhere and we record everywhere, and then we try to interpret how different regions are connected. And we're forced basically to use assumptions about the specific form of the response. So we have an explosion of comparisons. The most common way that this is addressed is people assume that there's a characteristic response shape, such as the N1, N2 shape seen in cortical evoked potentials, and think that every response should be parameterized in that context. I'll show you in a moment why we think that is not the best approach. One approach that has been used that we think is a reasonable way to go about doing this is something we call the hypothesis pre-selected approach. And that's where you have information either from historical neuroscience, from stroke studies, from fMRI, where we assume that two areas are interacting with one another. And so we examine just two areas to begin with. And we call this approach the hypothesis pre-selected paradigm. The way that I'm going to begin my studies and the way that this basis profile curve technique was developed is to examine data within the convergent paradigm. So here's a real world data from one of my epilepsy patients where we implanted electrodes in and around the temporal lobe. And here you see a blow up of the temporal lobe of this patient seen in coronal section. They happen to have a lesion in this area. We implanted electrodes both on the lateral convexity of the brain and also on the underside of the temporal lobe. Now, we used the following paradigm. Electrical currents were delivered in a biphasic way so as to preserve total charge delivered, and we allowed approximately 3 to 7 seconds between each pulse. We then record the responses to about 10 stimulations in a row at this site that you see here in red. We then can average these responses, and we call this average response the cortical evoked potential. And what you'll notice from the average of these responses is that there's nothing like the quote N1, N2 shape in the cortical evoked potential. So we couldn't parameterize our data using just the characteristic voltage at some delay compared with the stimulation time. We can then examine stimulation at a different site, as you see here, and look at the average from that site. And here you actually do see something similar to the N1, N2 response. I've zeroed out the data for the first 15 milliseconds following the response, but you can see that there is something like that characteristic response. Now, what we may ask is because the shape of these two responses that we measure here at this red site, which happens to be on the parahippocampal gyrus, are so different, does that difference in the shape of the voltage response tell us about fundamentally different types of connectivity between this parahippocampal gyrus site in red and this site in the superior temporal gyrus in green, and the site in the fusiform gyrus and the inferior frontal gyrus in purple? So one thing we can do is we can ask, well, are there general types of connectivity between the seed site that we picked to make all of our measurements in the convergent paradigm here on the parahippocampal gyrus, which you see circled in red, and many other sites from a grid of electrodes and strips of electrodes placed around the surface of this patient's brain? And what we do is we plot the average response, that cortical evoked potential, at the location between the two electrode sites that were stimulated. So you can see we stimulated in a pairwise fashion, and we see 41 different cortical evoke potentials plotted at the site where the stimulation took place. And it's important to keep in mind here that we're not measuring responses at any of these different sites. So this doesn't reduce to the types of things that we're used to looking at, say, spatial responses in PCA or other types of clustering techniques. But we're actually only measuring at this one particular site, and we're just plotting the response at this site at the place where we stimulated. So we can get some spatial representation of how this parahippocampal gyrus site may be responding to stimulation in other areas. And what we see if we look at these responses is that there are clusters of shapes that emerge visually. So we see this shape that is present in the superior temporal gyrus here, and it may look like some of these responses are actually superior to the Sylvian fissure, but that's because one of the electrodes was sitting on or below the Sylvian fissure in each of these cases in the green cluster. And we see a fundamentally different shape here in this orange cluster, and a different shape here at the stimulation pair at the purple site, corresponding to the site that I showed you in the previous slide. So these clusters emerge visually, and we can ask ourselves, is there a way we can identify these algorithmically? So for those of you that are used to standard clustering techniques or standard component decomposition techniques, the standard approaches simply don't apply. Namely, the fact that we're always measuring from the same site means that responses of similar shape but flipped polarity or flipped sign should never be clustered with one another, because we're only measuring from one site. And since it's not a source localization problem, we really should be clustering positive and negative responses differently. We also should allow some overlap in shape, because there may be elements of a response that, let's say, are preserved. For example, the characteristic shape from an input to a specific class of interneurons, followed by something very different, namely the response that you might have from a set of pyramidal neurons in some population of neurons. And what we would really like is a shape of each of these canonical responses, like I showed you before in those visually apparent clusters, along with relative weight and significance of that shape that we can plot on the location of stimulation. And we also want a set of insignificant stimulation sites. The approach that we've used to do this is we examine the data in a hierarchical fashion. So we can imagine first, we're just measuring from one particular site, and we look at how stimulations that are repeated in a given stimulation pair, which is what you see here in A, are related to one another. So we can take one response, and that's shown here in black. It's actually that top response from the left shaded or peach colored shaded region here. And we take the single trial response that we have, and we compare it to all other responses from that same site. And we repeat this process, comparing responses from stimulation at that site to all other responses, but never comparing a response to itself. And we can generate a set of projections, and if we just take the dot product of these two, we can generate a set of projections here and look at that distribution versus 0. So here we have what we call group 11, and that's basically a site. And because we have repeated stimulations at one particular site, we say that that set of repeated stimulations is a group. And then we try to basically leverage a hierarchical structure from that. So we have stimulations into, stimulation events that happen, and we project those into stimulations from the same site, and we get a distribution that you see here, where it's called group 11 self-projections. But then we also take this group and we do something very similar by projecting it into the responses that were elicited from stimulations at another site. In this site, we say group 23. And each of these, you can see here, is significantly different from 0. And in fact, what we have is we have a set of these projections for every pair of clusters, where we have a cluster. I apologize, cluster wasn't the right word. For every group of similar stimulation responses that happen because we stimulated the same site over and over, and we compare that to another group where stimulations happen from a different site, and we stimulated that site over and over and over. We look for similarities in those. And with that, we generate this projection matrix that we call S here. And that's really a set of responses that reflect all of the similarities between stimulation groups. And we generate a significance matrix from that. And that significance matrix represents T values for how different each set in that matrix of sets S is from 0. And then everything that's below 0, we set to 0, and that gives us the ability to do non-negative matrix factorization. And the rationale for setting that to 0 is that if there is a strong correlation in responses that come from 2 different stimulation sites, but that correlation is flipped, then those things should wind up, or those responses should wind up in different characteristic response shapes. And then we take that matrix of significances and decompose it using non-negative matrix factorization. And then we calculate the similarity within the clusters that emerge by looking at basically the inner product of the matrices that we extract from the non-negative matrix factorization. For those of you that are interested in it, there will be a link at the end of the talk to a paper that describes all of the mathematics in detail and has sets of original scripts you can use. And of course, we're also presenting an original package developed by Dr. Hermes here at Mayo Clinic to perform this automatically with your own data. So we look at the final matrix that's generated in this non-negative matrix factorization, which here was reduced to 3 rows, and the elements of each row tell us what stimulation groups are similar to one another. And then we use something called linear kernel PCA to extract canonical shapes from sets of stimulation groups that are clustered with one another. And that process is what we call basis profile curve identification. So the results of the basis profile curve identification, for example, are 3 sets of basis profile curves. One, as you see here in green, has initial positive deflection, it returns to 0. The second that you see here in purple has a negative deflection followed by a positive deflection. And the third that you see here in orange has a negative deflection that then returns to 0. And we can look at these responses scaled at the sites of stimulation that produced the responses that we measured. So it reproduces the clusters that we saw visually in areas that elicit a similar response down here at our measurement site, except that now they're all parameterized in terms of a single shape, and we measure the relative strength of projection of that shape into this cluster that you see here in green, and a different shape into this cluster that you see here in orange, and a different shape at this site that is produced that's really its own cluster here in purple. And the algorithm itself can identify, basically it identifies a number of clusters that is optimal based upon how related the shapes are to one another. And that's a parameter that you can tune if you wish, although the automated settings we have on that are, I think, reasonable from the start. But the parameter that we wound up with here in our example of 3 clusters actually maximally captures the portion of variance explained across all included trials from the sets of responses that are brought into the process at the end, because some of these sites that we measured here are relatively insignificant. And so because they did not produce a significant reproducible response shape, we feel that it's not relevant to include those in our estimate of what the portion of variance explained was. And that really results in the fact that sometimes you measure from one brain site and you stimulate another that's not meaningfully connected, and you don't get a reliable deflection in the voltage in response to that stimulation. So the take-home points from this work is that a convergent paradigm yields interpretable motifs in connectivity. This algorithm that we developed to identify basis profile curves satisfies a clear set of constraints, and it gives us something concrete that we can use to understand the responses that are produced at a given site by stimulating elsewhere in the brain. That's these basis profile curves. So I hope that this method will be useful for those that trial the technique, and I very much appreciate the opportunity to share this with you. This is work, again, that I did with Klaus Mueller from Berlin and Dora Hermes from here in Minnesota. Thank you for your time. Have a nice day.
Thank you. Are there any questions about this? So please raise your hand if there's any questions, or just pop it in the chat. I see that there are some people who joined a little bit later. So we'll be walking through the tutorial in a step-by-step basis so that everybody can follow. Okay. So I'm sharing the Jupyter notebook now that shows the package that was that Kai just talked about, to identify basis profile curves to understand the effects of electrical stimulation in human brain networks. And one of the things that this is, we're going to walk through one example data set now and see how this works. This effort is part of an NIH grant for which we are currently collecting many of the same data sets, and we'll be sharing those openly. So we hope that with these tools, now there's an example data set here, but we'll be sharing many other data sets too, where the same method can be applied. So this paper, here's a brief abstract, is is currently under review. It has already MATLAB code available with the paper, but here we are sharing a Jupyter notebook such that it's more visible what it does and how that everybody can use it, even if you don't have a MATLAB license. So before we're going to get started with the practical session, if you have any technical issues, please put them in the chat and just make sure they say technical colon, because I wanted to mention that Harvey Wang and Tal Palatia are here also today to help field any of the technical questions. I'll be taking more of the content-wise questions, but if you have any technical questions, please feel free to put them in the chat to make sure that everybody can walk through if they want to. So the first part was hopefully shared ahead of time, but I don't know if everybody had time to do that. But in order to get started with this tutorial, you have to make sure that you have your Python environment and packages up and running. So in the Jupyter notebook here, you can see on the top right that I have the BPC, basis profile curve environment set up and running. The simplest way to do that is to uncomment this section and then just run this cell that should automatically set up your environment. You may have to refresh your browser window in order to make sure that, and then you go to kernel, change kernel, and make sure you have the BPC environment. If all that is done correctly, you should be able to import the Python packages as I've done here. Now, this Jupyter notebook works with the data the example CCP dataset or cortical Evoke potential dataset that is organized according to the brain imaging data structure. It was made available on OpenNeuro. So you can see here, this is you can go here and download the data set, which I've already done. And the nice thing about the brain imaging data structure is that it's described in a very standard way. And all the naming conventions are standardized. So you can see here that if we click on subject 1, there's just one subject of an example, and IEG01, we can see that there's only an intracranial EEG format. Sometimes people share a lot of functional MRI data on here, EEG data, MEG data, and here you can see the format for intracranial data. And for example, we can already rapidly view the events, for example, and we can see that there are that there's an onset of events in seconds, and you can see here which electrodes are stimulated during this electrical stimulation paradigm. And since the format is in such a standard way, we can is standardized, and there are tools being developed for analyzing these data. This is what we use for the for downloading for this workshop. Okay, I just saw that someone still missed the link, and I will just make sure that the code is. So I posted it again for a little bit earlier. Are there any other questions about this? Okay. So after you have the data somewhere on your computer, then you have to make sure that you set the path to that. So for me, the data are located in this path, and then I make sure that the path is set. Just to check whether everything is working correctly, whether it's loaded correctly, you should be saying when you run this particular cell, you should see that there's one subject, one session, one run, as we also saw in the example here. There's one subject, one session, and one run of data organized in the MEF3 format, because that allows for parallel computing. Okay, we don't actually use data. We don't actually need DataLad to download the data. You can just click on this download button, and then it should download. Thank you for these questions. So. And the way in which this works is this is using code from PyBits. PyBits initializes the bits layout such that we can now automatically start pulling information from this brain imaging data structure. So another question that's coming up is we cannot start the interactive notebook. How do we do that? So if you have Anaconda running, for example, it can yes, you have to clone you have to make sure you have the repository downloaded or cloned, and then you have to make sure that, and the simplest way that I've done that is make sure that I have Anaconda installed, and I click on a Jupyter notebook, and then it just runs from the browser. Okay. So here, after we have our bids layout, I will we can see we have to basically put in the subject number, session number, task, and the space in which the electrodes are, which is currently the MNI space that we want to analyze. So you can either look that up here in the data set. You can see here what the space should be. Subject label should be 01. Session label should be IEG 01. The space label should be, you can see that right here. And then you just put in this information, which is already set standardly for this particular subject. And then and we can download the file names and metadata for the analyses. So what we do now is that we retrieve all the file names so that we can automatically load these in, load these here. So we make sure that, so the brain imaging data structure has several aspects in the metadata that are important to consider here. And I will walk over this because a lot more people will be sharing data in the brain imaging data structure, given the fact that it's just very simple, straightforward, human readable and machine readable to use. So I will be walking through this while people are having, while people can download it, just to explain a little bit what's in the brain imaging data structure. So here you can see that, for example, we have a data JSON file, which has standard information on the sampling frequency. And so everybody can access the sampling frequency in the same way. And we can do that for EEG datasets, intracranial EEG datasets, MEG datasets, etc. We can load the channel information that were recorded on the amplifier. We can load the electrode positions data information, which is different. So these are the hardware contacts that were that were implanted. So information about the XYZ positions, what type of material they were from, how large they were, whether it was a stereo EEG or an electrocardiography probe, and then of course the events. And so once we have all this information, we can use the pandas data frame to quickly test if this is running correctly. And then, for example, here in the channels.tsv, you can see that there's, for example, the electrode name, electrode type. You can see that is electrocardiography here, so surface electrodes. Units should be microvolts. There's a low pass filter, a high pass filter, hardware filters that are standardly applied in these for these data. It's an intracranial electrode, and then there's the sampling frequency also here. We also annotate for each channel whether it is good or bad, because sometimes in intracranial recordings, for example, some electrodes can be bad due to artifacts, due to noise, due to the connection, and all these data are stored in a standardized way in bits, such that you can look up in the brain imaging data structure what the standard should be. And then the events. For the events, you can see here the onsets in seconds, the duration in seconds as well. So you can see that the duration of the electrical stimulation in this case is 200 microseconds. And you can see that the trial type is electrical stimulation. We annotate the sample that was started, which electrodes were stimulated, which electrodes numbers this was, what the current was, etc., and whether the trial is good or bad, which is important because you want to include any trials that potentially had any more noise. And then lastly, you should be able to successfully load the electrodes.tsv file, which has, again, the name that should match a name also in the recordings, and the XYZ positions that are in this case in MNI 152 space, such that we can standardly plot them on a standard brain and get information. You can also see that in this case it was a subject who was who had electrodes on the left hemisphere. And then you can see that what we overloaded here in the bit structure, you can always add more information as long as there is an electrodes.json file. There is someone, I don't know. I have a brief question for the monitor. Someone requesting remote control of the screen. I think I'll have to click decline.
Yes, please click decline.
Thank you. So one of the things that we overload here in the brain imaging data structure, because we can add additional information that is not specified in bits, as long as we make sure that there is a JSON file that describes the columns that are entered here. So what we do here as well is we make sure that we add the labels that were most common from free servers, such as the in this case the Stereo Atlas, where you can see that this electrode was localized on the temporal pole, for example, or the inferior temporal gyrus. So we add as much information as possible so that other people can actually use the data. And this is one of the first examples that we'll be sharing with this larger set. But we hope that in a few years we'll be able, like at the end of this grant, we'll be making more than 100 data sets available in the same format and in the same structures. So there is a question, are the time series pre-processed in any way? No. So the data that we're sharing here, the data that the brain imaging data, there are several extensions to extend the brain imaging data structure to pre-processed files. But those should be shared in derivatives. And there are several efforts where we're extending the, we're specifically extending the brain imaging data structure for electrophysiology. And there's always comments there. Welcome to see where you would like us to take that. For the purpose of this workshop, we really wanted to make sure that we share the entire pre-processing pipeline. Therefore, that is a little bit tweaked to this data set, but should work in a more general setting for intracranial EEG data, such that people can go back and see what we did to optimize the data. One of the things that, in terms of the pre-processing, that we did add in this data set is the status for the different events. So we reviewed all the, Gabriella Ojeda, my research assistant, reviews all the data and looks at whether there's any artifact in these trials or not, which is fantastic, because then we can actually say whether the data have large artifacts or not. If it's labeled as good, we can use it for analyses. If it's labeled as bad, and that means that there was an artifact on the multiple channels, and we do not want to use that. So there are things that could either be seen as annotations or pre-processing steps, kind of looking for good or bad trials that we have added in the data set to make it most useful for other people. Okay, so we'll be going over the pre-processing in, as Harvey mentioned in the chat, also in section 3 of the notebook. Are there any questions up to here? I don't see any more questions or raised hands. So what we will be able to do now is we can render the brain surface. And you can see here the locations of the electrodes with the names. So we can hold on, you can just use your mouse to scroll around and see where the electrodes were implanted. You can see that there was a large grid implanted here. The LTG left temporal grid from LTG number 1 is over here. LTG number 48 is over here. There were some subtemporal strips, LPS, LMS, LAS, and some electrodes on the anterior temporal lobe. You can also see here that if you scroll a little bit to the side, or if you scroll around it, there were also a few depth electrodes, a few depth probes implanted right there at the back. Another important thing to note is that some of the electrodes are marked in gray in particular, and those electrodes, those are the channels that were marked as bad. So those did not have a good signal throughout the recordings. So I hope people have been able to bring up this figure, because we'll go, because what we'll be able to do next is I'll leave this open, and we'll be able to go back and forth to look at several electrodes that we're interested in, and look at the inputs from when we stimulated many of the other electrodes into this particular, into a particular electrode of interest. And so this is we'll go through the pre-processing that we do in this case to the data. And so we have to pick an electrode of interest. And one of my favorite ones that's a good example to start with is electrode LMS3, that you can see here, is located right here on the parahippocampal gyrus. This is a good one to start with if you want to test the code to see whether everything runs. At the end of the notebook, I make some suggestions of other questions. We can run through another. We can go through another couple of examples from one of the depth electrodes and one of the grid electrodes to see how things are and how things change. And that's the interactive part of the workshop, is where you can try things out and see how that works and then ask questions. Okay, Jupyter install is having some technical issues. So I think there are some people having a little bit of trouble with the Jupyter installation. So I will give it a minute to make sure that people can install it. So okay. So we'll start with electrode LMS2. And then since we have everything in bits, we can extract the relevant information to pre-process the data, such as, for example, the sampling frequency, which is extremely relevant. We here can now say, because we have everything standardized, we only want to include the good trials that do not have any artifacts. So we want to have trials that had electrical stimulation, that had a status good, and also, we only want to include the trials where the electrode of interest itself was not stimulated. So we want to exclude that. So for what we do here when we pre-process the data is that we apply a common average re-referencing, but only for those channels that had the least variance. So we do not want to have electrodes that had artifacts, etc. And here we just have a basic parsing of what was good or bad. And since everything is standard in this structure, will be this is where we're going from. And you can, for example, see here as output data are collected at 2000 Hertz. And now, while I talk through the calculation of the convergent paradigm briefly, if people have things up and running by this time, we should start the pre-processing section. We should start running this script because it takes a little while. It takes a few minutes for me, but it depends on the computer that you have. You can see here that it loads all the trials, applies the pre-processing steps, and calculates the convergent matrix. So while we are doing this calculation, you can see here in the star that it's running. I just quickly want to reiterate some of the parts from Kai's talk, and feel free to ask questions here about the convergent framework as well, the convergent paradigm. So in the convergent paradigm, we specifically look at inputs from many different areas. So when we stimulate many different sites on one particular area that we have now selected as this particular ventral temporal electrode. If we look at the data in this way, and we have stimulated many other electrodes, different inputs and different shapes of the response curves have a specifically different interpretation because we only measure in one site. And for example, a superficial or a deeper input will have a different effect on the measurement site and may, for example, reverse polarity. So looking at these cortical Evoke potentials in this convergent paradigm allows us to have to separate these different inputs. And that's what we wanted to do here, is we wanted to have a tool, a mathematical tool, clustering technique, that separates particular inputs to see how those different sites cluster together. That's different from the divergent paradigm, where you stimulate only one site and look at the effects on many other regions. And then there's different ways, of course, to re-reference the data. While the pre-processing is going, it's actually done now. This is something that is pretty specific to what we do to our data, but will probably work in many different settings, is we do a common average re-referencing, and which excludes channels with high variance in a later time section or in a further time section for 10 to 100 milliseconds after stimulation onset, such as channels with artifacts, or channel evoked responses. We do not want to introduce those on all other channels, so we remove that. So that is working, and that is what's going on in this section is the pre-processing of the data, and also clipping it in terms of the convergent matrix V, where we see all the inputs from all the individual trials arriving in this particular electrode. Okay. So now we can plot this matrix. So here you can see on the x-axis, you can see time before and after electrical stimulation, with the electrical stimulation happening at point 0, and in all the rows here, you see all the individual trials when another pair of electrodes was stimulated. So what you can see here, and in color, you can see here, for example, that when these paired numbers were stimulated during these trials, you can see a large negative deflection. And here you can, for example, see a positive deflection. And you can actually see, if you look carefully, that there's 2 negative deflections in these trials, whereas here for this particular pair, there's only a later response. Now, one of the additional pre-processing steps that you see in place here on the right is that we did a baseline correction. So for every individual trial, we subtract the average of the minus 500 to minus 50 millisecond before the trial, such that the trials are all centered around 0 before stimulation starts. Which you can see here that this offset that is here on some of these trials is removed in the with baseline correction, because we're not interested in looking at these offsets. We're really interested in the evoked responses. Are there any questions about the pre-processing part? So there's one question. Using the common average will change the response shape depending on which electrodes were used, correct? Yeah. So that's one of the reasons why we, so common average, if we take the common average of all the channels, and we wouldn't take only the percentage of channels as we do here, the 75% channels that had the least variance, then it might change the response shape. So if we were to include channels that have a large response, we would, by common average rereferencing, introduce that response in many other channels. That's correct. That's why we only take into calculating the common average, we only take the channels that had the smallest variance. And then we do not introduce a response in other channels. So you can see here that the responses are relatively well contained, and it's not like because there's a large response here that's not introduced in many of the other channels as well. So that's why we do this particular common average rereferencing for the CCP data, where we only take the average of the channels with the least responses. Does that make sense? Okay, yeah, but this is always something that is very necessary to really carefully check. And so we set our threshold at 75%, so we only take the 75% of channels with the least variance. And you could, for example, if you have a lot of responses, dependent on the data type, if you use any other data type, we have here these particular CCP data where that's generally a good ballpark. And if you have other data, there maybe that may differ. Okay, so now we are ready to start calculating the basis profile curves. And one of the things that we have to think about is within these basis profile curves and this convergent paradigm is which time period are we interested in? And for example, here you see that most of the response shapes and the largest responses are visible from after stimulation to about 1 second. After about a second, the responses are basically over. So what we do here is we take from 15 milliseconds after the onset of electrical stimulation to 1 second. That's the period that we take to calculate our basis profile curves. And so in order to continue with the calculation of the BPC code, what this uses also is the convergent matrix V that we have visualized here, a time vector to know what the times are here. And then we have a pandas data frame that's called pair types that we could quickly visualize. If I add an extra cell, just that will be constructed actually in a subsequent section right here. Here, pair types is constructed, where you can see how these trials are grouped into different stimulation pairs, because these are all the single trials. So let's have a brief look after we calculate. So here you can see the significance matrix, but before we go there, let's have a brief look at pair types, where you can see that, for example, that during these trial numbers, which are here on the y-axis, we stimulated electrode elements 3 and 4, which is electrode numbers 6 and 7, 5 times. We generally stimulate every pair about 10 times, but in some cases there are some, as I mentioned, there are some events that are removed due to the fact that there are artifacts in this case, and so we remove those. So here you can see that in total there were 38 pairs of electrodes that were stimulated, and these are the trial numbers that index into the events that were stimulated. And what we then do here is we calculate these cross projections that Kai just mentioned in his talk, where for every pair of electrodes, the trials are projected into the pairs of the same type, which is the diagonal, and into the pairs of other types, which are the off-diagonal elements. And so you can see that there are several subgroups where, for example, when we stimulate pair number 11, those trials are very similar to when we stimulate pair number 1, 2, 3, 4, or 5, etc. So this matrix is important to just always check and just see what is there, and you can kind of get an initial feel for how trials cluster together. And you can see that some trials cluster together with others, whereas you can see some others clustering together. And this is what we've entered in the non-negative matrix factorization to find sites that cluster. So we run this pair. We're in the cell, and you can see that this through non-negative matrix factorization, we reduce this to a number of pair types that cluster together, and then every electrode stimulation pair is only assigned to one of those. So here we take a winner-take-all approach, where every stimulation pair is only assigned to one of these clusters. You can see here that we get 3 clusters. And we'll go through this relatively quickly and come back to this if there's any questions. If a pair is similar, does it mean the same effect occurs independent of the stimulation site? Yes, that's correct. So if you have so you can see here that, for example, these stimulation pairs, 1 and 2, elicited a very similar response, and they clustered into the same basis profile curve. And so here we can quickly see what these basis profile curves look like. And for example, you can see that the first one, the blue one, just had a late negative response. And so when we stimulated these pair numbers of electrodes, they all produced an effect that was very similar to the blue curve. You can see that for example, pairs number 2 produced this very classical N1 and 2 response that has received most attention for cortical Evoke potentials. So an early negative response followed by a later negative response. And that's what people have mostly used to look at these as cortical Evoke potential, is people have generally looked at the N1 component and see how that defines connectivity between areas. But what you can see here is that there's actually several different types of shapes. Not all electrodes only elicit this N1 and 2, but there are some that also, when we stimulate some groups of electrodes that cluster together, that only elicit like the second negative response, and some that elicit this positive response. And so using this clustering technique, these responses are specifically grouped together. Are there any other questions about this? If not, I will go look at the spatial representation here. Okay, the three curves are the plots of the three clusters. Yes, so these sorry. So what you saw here, these are the temporal waveforms of these three clusters. So this is the three clusters that we get for this particular pair. And here we can see when we run the spatial representation part, you can see the spatial representation of these three clusters. You can see here that this particular cluster that was located on the superior temporal gyrus resulted in this positive curve right here. You can see that the measured site is highlighted in black. And when these green electrodes were large and stimulated, those resulted in this N1 and 2 response. When those blue sites were stimulated, especially when they're large, so you can see that these are smaller. So these were, when they're very small, they probably wouldn't reach significance. But when they're smaller, they elicit only the second negative, slower response. So you can see how different electrode pairs group together. So there's another question. Does the curve form tell anything about the inhibitory or excitatory effect? So that's one of the things that we will now be able to figure out, because these are slower responses. While most attention has generally been given to this early response that is thought to reflect the direct cortical connections through, and has been even correlated with some, in some ways, with white matter connections. These slower responses are relatively unexplored. And that's partially because there's been very little attention for those. Because of the focus on the particular N1 and 2 shape. So with this identification, we will now be able to study whether those are more excitatory or inhibitory. What seems clear is that there's a different pathway to which they affect the measured electrode, but exactly whether that's excitatory or inhibitory, this method will set the basis for being able to figure that out. Another question, is there a way to tell which direction are the clusters in relation to the cortical surface? Orange and green are opposite at 200 milliseconds. So we are always measuring in one electrode. And so even though these clusters are in opposite directions, we here stimulate this pair for, you can see here that what's interesting is that if we look at this measured electrode, the green electrode here is positioned between two white dots. That is because this pair is when we stimulate across these two white dots. So we stimulate these and we measure responses here, or we stimulate these electrodes and we measure responses here. Or we stimulate across these electrodes or across these electrodes. So it's not just that we stimulate in the opposite direction and we get an opposite response. These electrodes have a very, in terms of brain space, a very different type of a very different way in which they project to this particular area. And also, for example, in terms of the blue, which is opposite again from the orange cluster, those have a different shape. So we can now start grouping these together. Okay, what areas does the left superior temporal gyroscope correspond to? Does it differ by diagnostic group? So in this case, the patient had a tumor that was located between these depth probes. So it doesn't seem to, and with epilepsy around that region. But these particular electrodes it was not a particular diagnostic group of electrodes. It really allows us to pull out clusters of connected electrodes. So, oh, sorry. Yes. So we are now able to figure out more of the connected, how a group of or group of electrodes or a particular brain region clusters together in terms of affecting this particular area. So I don't think it's particular. It might be diagnostic in some cases, but we would have to look at particular types of epilepsy and whether that differs from typical basis profile curves compared to when there's disease from a particular area. And so I hope that answers that question. So thank you for all the questions, by the way, I really appreciate it. So at the measured site, is the current going up or down the 6 layers? Okay, so so that's what we think. So we always measure, in this case, since it's ECOG, we measure on the cortical surface. One of the hypotheses could potentially, since we are on the cortical surface here, and we have electrodes that have a particular positive or negative response, that could indicate that there's a particular they project to more deeper layers or more superficial layers, or that elicit a different response. Yes, that's one of our interpretations, but exactly how that would what the neurobiology of the underlying response would be, that's what we will be able to figure out now. And that's why we wanted to make this technique available and share it with data such that we can look at this from different ways. Because what we mentioned in the beginning is that it's mostly everybody looks at just general connectivity in terms of maybe the N1 and the latency and the locations for that. But in terms of the projection sites, this technique opens a different way of looking at the data and looking at those representations. And for example, we're giving one example now about the about one ventral temporal site that is on the on this particular area in the on the parahippocampal gyrus. And now where I'm interested in the in visual processing and processing in ventral temporal cortex, but in this particular data set there are many other sites that might potentially be interesting. So this will you publish data with more subjects? Yes, we will be for this grant we will be making a set of at least 100 subjects available in the next 4 years is the plan. So that's one of the reasons why we want to make sure that we share these data. We are in the process now of all organizing these data in the brain imaging data structure in the same way as these data are organized. So any tools that will be developed for this example data set, we will be releasing as the data come in and as we are starting to curate more and we have more sets available. There will be more of these types of data. Much more of these types of data that will be released, and we're hoping that people will use these tools and these data to find new questions, because you can only do so much. Could it be that responses are actually composed of a linear summation of multiple component curves, with each component having a more direct physiological interpretation? If so, how might the method be modified to quantify these components? So the So it would be possible. So we've seen that, we've seen in some other data sets that we see that there are, for example, that are sites that cluster together, and one having, for example, some of these early responses, and another not. So, for example, you see some of the sites have these early responses, and so in the future, what we think is possible right now, these are all clustered into, these are basically, that would be, we take a winner-take-all approach where we assign those, but you could imagine an approach where we do not take a winner-take-all approach, but we would say if a pair contributes to 2 basis profile curves and has a 50% contribution to one and a 50% contribution to another, it might be possible that it contributes to both. So you could change, in particular, the winner-take-all step in order to let one basis profile curve contribute to, or one stimulation pair contribute to multiple basis profile curves. So I hope so that's the step that could be modified in order to calculate that. The other question was that how can each component have a more direct physiological interpretation? I think this method, in terms of a direct physiological interpretation, having the time series assigned to clusters of electrodes, or of how they affect a particular area, will allow you to, in the future, understand more what the physiological interpretation is. We often see, in terms of our connectivity analysis, we pick one feature, such as, for example, the N1, or such as Granger causality for one particular frequency or one particular component, to study these connections and network interactions. And what we want to do here is we want to push that away a little bit, or like enrich that framework with looking at how different clusters drive one area. And I think we have time to look at, for example, another example electrode, and give people a little bit of time to run through the code and see whether there's any other questions. So that you can see how another electrode, that's, for example, one of the electrodes that is on the anterior temporal lobe, how that looks different. So another example that would be a good one. And one of the things that that I would like to mention here is that these are some examples that I know have reliable BPCs, and because these measured electrodes that I'm selecting here as examples did have a reliable Evoke response for some of the other stimulation pairs. With ECOG and Stereo Eeg, we measure sparsely in many different areas. So it is possible that we measure from an electrode and we just did not stimulate a well-connected site. So we also will have pairs of stimulation, or measured electrodes, where we do not have a lot of signal. So if you only throw noise into your analyses, noise out is what you get. So I know that this electrode on that's LAG 58, that's located right here on the temporal pole, also has a nice set of basis profile curves. So while we are calculating the convergent matrix, we'll plot it afterwards. You can see that this electrode will also have a few nice curves, and that's why we wanted to make sure we visualize this convergent matrix, such that you can see whether you have any reliable responses. Because if this only looks like noise, then you either had an electrode where you just didn't measure many Evoke potentials, or your data are just, maybe you didn't stimulate with the correct, with enough currents to induce any of these Evoke responses. The stimulation trials that we picked for this particular data set, stimulating at for 200 microseconds with 6 milliamps for an ECOG data set, is relatively typical in terms of amplitudes and size. So it should be similar. You should get similar outputs with other data sets if they would be formatted in the same way. Just going to take a brief sip of water. So. Okay. So so there's another question. You mentioned that you censored some electrodes trials after visual inspection. Could you elaborate on the criteria for censoring data? Yes, this is a really important question. So before we do any analysis, before we look at any of the basis profile curves, any of these things, we look through the data visually, and anytime, sometimes the subjects move their head, like they move their head a little bit, and then there's noise on all channels. Sometimes and then those are the trials that we remove. So if on all the channels at the same time there's a sharp spike or an artifact in the data, that's not something we want to take into account, or we that's not something we want to take into account or we want to analyze. So that's one of the artifacts we look at. Another thing that we look at is if there is an epileptic spike that is really large, that is happening across all data sets. That's often something we do list or we do look into, and we want to remove that from the data as well, or we want to label it at least to understand what was going on. So those are the trials that are rejected. Another thing that we do that was asked about was the channels that were removed. Sometimes there's a channel series, as in this case, there was a channel set like this strip. Just was noisy, had a lot of 60 hertz line noise. That's probably because there is either something in the cable or in the connection that is not functioning as well. That's a pretty well-known issue for ECOG, that we sometimes have these sets of electrodes that just have a lot of 60 hertz line noise. So that's always something we throw out of the data. We can't use that. So those are the channels that were marked, the channels and the trials that were marked as bad. So now that we have calculated the conversion matrix, we can plot it here, and you can see that it looks different from what you saw on in the other set. You can see that there's, in this case, there's again some electrodes that show these two negative responses in bright blue, with like some positive responses intermediately, and maybe here on the bottom a few that have positive responses. You can see that it's different from what the other electrode shows. And so one of the ways also in which why we plot this conversion matrix, that if there were some trials in here, for example, that still had those artifacts on them, you might see like a bright yellow line crossing through in the middle. And so those are the trials. This is a nice way to visualize all trials for one electrode at the same time. I mean, we have about almost 80 measured electrodes in this case. So you would have to look at this for 80 electrodes to see whether you move bad trials for all electrodes. But this is basically for the electrode of interest that we're looking at. In this case, we get a pretty rapid view also, like whether there were any leftover artifacts in the data that would then trickle into our analyses. So were there any tasks or feedback from the participants during the stimulation? Did they have any visual illusions when stimulating the visual areas? So in general, we ask our participants during this particular task to just be awake, not fall asleep, and just watch TV or chat or just hang out in the hospital bed while we do the electrical stimulation. And we ask them to give us feedback on if they have any strange experiences at that time. So these are pretty higher order visual electrodes in the ventral temporal areas. So they did not have any visual illusions when we stimulated. The level of current and duration of the stimulation, it's a single pulse delivered for 200 microsecond duration with 6 milliamps, is too short to elicit, in this case, any visual illusions that we've seen in the about 30 cases that we've done now in higher order visual areas. However, if we move to motor cortex, for example, if we generally do not stimulate in the data that we're collecting now, we generally do not stimulate motor cortex. A collaborator of ours does generally do that, but we skip that because we do induce twitches in the hand or in the feet that subjects may find uncomfortable. So we generally skip the motor cortex electrodes, but they might, that's so sensitive that it might drive a response. Okay, and so we're going to choose the same time frame, for example, for calculating some of these basis profile curves, because again, that's kind of where we do see the main effects. And here's our significant matrix again, and we see some groups of electrodes, some things clustering together in these rows and columns. Then we do the non-negative matrix factorization, where we can see how many components we end up with for this particular pair. We see that we have 3 components, assign each stimulation pair subgroup to 1 cluster. And we look at the outputs. So we see we have 3 components. There's two stimulation pairs that came up in the first one, a whole set that came up in the second one, and then a few in the last. And then when we do the visualization, you can see that they have very different shapes that are automatically extracted from the data that we can now separate. And it's interesting that these orange and green curves are overall very similar, but there are slight differences. Here, the green one has an additional early component and is a little bit later here in the last negative deflection. Whereas again, there is also a positive response that is elicited in these electrodes. That is one of these responses that's not classically described in the literature that has emphasized most of the N1 and 2 responses. So there's many new things we can now start discovering from these data in a data-driven fashion. When we look at the inputs into this area, so this is the electrode that we selected now, we interestingly see a similar cluster projecting to this side as compared to the other ventral temporal side. And there's still a green, but it's now split up into this green and orange cluster that has a slightly different effect in the earliest time period. And we see inputs from the blue inputs from arising from this particular area. So we do see the spatial difference. We do see that this particular cluster of electrodes on the superior temporal gyrus both projects to 2 different sites, but sometimes can have a different effect dependent on if we allow it also to look at these very early responses here that occur around 30 milliseconds or so after stimulation onset. So this is another set where we can see something very similar. The manuscript has a list of good electrodes for the signal. So we do not have a list of good electrodes for the signal in particular that we assign in the manuscript. These are just a few examples, but this is the reason why it's important to dynamically and interactively explore this. So if you pick 3 electrodes that are side by side, do they have similar arriving these basis profile curves is a question that you can ask for different regions that are, for example, clustered in terms of these that we see here. For example, if we look at the data, we look at the electrode positions that are right here, the electrodes of TSV file. And the nice thing is that these things are both human and machine readable. And we see these Stereo labels as you could as well see here in the electrodes. You can here see, and I can show a little bit more. So you can see here, for example, that there are several electrodes on the inferior temporal gyrus, that you can then see whether all these electrodes on the inferior temporal gyrus that are assigned anatomically to or anatomically in the same region, do they also get similar inputs from other regions? And so we are starting to apply these methods now and extend these to larger areas. And how can we go from these basis profile curves that are identified on individual electrode level to a region? So that's the next step that we're working on now, is how do we take this kind of approach? This allows you already to look at one electrode that's, for example, involved in a particular function, such as motor function or visual perception. Then you can start exploring this throughout different sets of electrodes. And that's why we also have these labels there, so that people can see what is good and what are good electrodes that get good inputs. Another important question to ask is whether this method will also extend to Stereo Eeg. And the nice thing is that we have several Stereo Eeg electrodes in this same data set that were some of these depth probes. So if we, for example, look at LAD1, that is one of the electrodes you can see hidden right back there. One of these little 4 electrode depth electrodes, we can see how that works for a depth electrode, because we can use the exact same framework because we are all in the measured site. So statistically, we can compare all our inputs into this measured site. Still, the convergent framework applies here. And we can calculate all the inputs again into this region. And this takes only a few minutes. It just depends on how large the data set is. We're collecting data at 2000 Hertz for about 80 channels. So we're just loading the data in real time here. I hope people are able to walk through this as well and explore a little bit on this side. And if you're unable to run through the tutorial today, please feel free to afterwards also, if there are any issues with anything, we've pretty well tested it. But if you have any issues running it, this is the link to the code on our website on GitHub. If you have any issues and they seem to be there, please feel free to post any issues here, and we'll be able to help answer those questions as well. So I will put this link in the chat for anyone who's listed. And go back to, this is the data. Oh, sorry, I just, sorry, I didn't, let me do that again. So now everybody should have the links again to the GitHub page and to the so anyone who should be able to, if you just say you can download the code, then you should be able. This is the workshop that we're looking. This is the Jupyter notebook that we're looking at right now. If you have an issue or you think there's an error there, feel free to open an issue here, and we'll have a look at that and bug me if we're not responding. Here you can see that for the Stereo Eeg electrode, that it's very essential to do the baseline correction. You can see some electrodes here that have these large negative, kind of offsets in terms of the data. And after baseline correction, we basically have cleaned that up. So we can calculate the basis profile curves in the same way. For Stereo Eeg, there's a lot of electrodes that had similar responses, which is what we also see here, that many of these stimulation pairs elicit a similar response. And that makes sense because we are with the Stereo Eeg electrode, we're in the depth, and there are many electrodes here that are right above that likely elicit a very similar response in the electrode underneath. So we can here pick our window, and then calculate the matrix, which we did, and then do the basis profile curve identification again, and see how many components there will be for this particular cluster. Every pair gets assigned to one group, and we have several pairs for these basis profile curves, and we can visualize them. You can see that they look very different for these particular depth electrodes compared to the surface electrodes. So you can really see how important it is that we don't just rely on one component, such as an N1, that would generalize to all cases. However, one of the things that is interesting, that if we look at the depth, apparently we sometimes get these more positive responses rather than these negative ones on the surface. So you can see many of these details in those sites. You can see that, interestingly, this site at this particular depth, some electrodes that are right above elicit the blue basis profile curve, as you could see here in terms of shape, whereas there's a different shape again for these orange surrounding electrodes. So we can discover entirely new principles just based on the richness of these data, and look at the spatial representation. So in this case, we walked through, for these cases, we walked through two different electrode pairs. We walked through a particular time window of interest. And if anyone has completed this, it's interesting to go back to section 4.1, for example, and change the time interval over which the BPCs are calculated, and look how that affects the outputs. That's another thing that you can do. You can also, as we did, pick a different electrode and look at the various inputs. But you can imagine that if you're not interested in these particular early responses, and you only want to look at the longer time scales and see how those projections differ, you would only look, for example, from 100 milliseconds or 50 milliseconds onwards, and then you would ignore these earlier responses. So the method can be adapted, and you can basically, with this Jupyter notebook, you are allowed to change it in many different ways to you can use it in many ways, and it allows you to look at the data in many different ways. How do we access the visualization figure? Okay, so when you run this this cell, it will just pop up. The visualization figure will just pop up automatically in a new browser window. So it just, you don't have to do anything. It will just pop up as HTML in a new browser window. Take snapshots, etc. What's a nice feature here is that you can, if you're only interested in more surface electrodes, you may want to make it less shaded. If you are more, if you are interested in seeing the depth electrodes, of course, you want to make the brain a little bit more transparent. So there are small changes that you can make here. What you can also see here in this case is that there are some gray electrodes that are positioned between these white ones. You can see, for example, here there's 2 white electrodes, and these grays are the stimulation pairs that were non-significant. So you see these little gray dots that did not contribute to any of the BPCs and didn't elicit any responses. And that's important because those, or didn't cluster into any of those responses. So you can see here which electrodes pairs were stimulated in a dynamic way. You can see, for example, that in this case, we took the approach to stimulate across the pairs in this direction. And that is because the pairs, because of the way in which we stimulate is just from LTG 25 to 26 to 27, etc. So from so here we go from I'll zoom out a little bit. Well, we stimulate from there to there to there, but we did not have the time to stimulate between these 2 electrode pairs in this direction. So you can, in addition to seeing the outputs from the basis profile curve identification, you can also get a very clear look at the data in a 3-dimensional view rather than just looking at a long list of what was of like pairs that we stimulated. You can just see it in a dynamic way and look at it. And for example, you can see that we didn't stimulate these electrodes, and that so we cannot actually see what the inputs from the stimulation pairs would be because we didn't stimulate those. And that's very important because of the sparse nature of our measurements and the fact that we only can do so much, or we have noise in our data. And this allows you to see that at the same time, we didn't blank out everything and just only emphasize what was significant, but you can see what was significant, what we did and what was not significant, and what we didn't stimulate. So you can see all 3 of these responses and get a full view of the data set. In addition to getting the nice outputs, it allows you to really get a core sense of the extent of our measurements, which is really nice for ECOG, but at the same time has its core limitations. So, okay. Are there any more questions about this? I think we have another 10 minutes or so. Okay.
If there aren't more questions, it's okay to disband the room a little bit early. I mean, you've already been talking for a very long time, so I commend you.
Thank you so much. Were you able to run through it?
I wasn't over here. I've been jumping from room to room.
Oh, nice. Yeah, no, this has been fantastic, and there's been a lot of questions.
Lots of questions and interaction. I'm really pleased with it. We have one more question down here, I think.
Okay, generally, do you think these Evoke potentials primarily measure structural connectivity or also informs functional connectivity? So I think this is actually extremely relevant for looking at functional connectivity as well, because because what we see here is when we electrically stimulate, we have a particular effect on one area. Now, that effect might be either direct or indirect. So we could have a direct effect, which, if we looked at these N1 responses, they have been tied to structural connectivity, but all these other later responses are probably more of a functional part related to indirect connections as well. So if you have an indirect connection that goes, for example, through the thalamus or through another third site, then that likely affects these later responses that we see after about 50 milliseconds. So I think that it's very important for functional connectivity, how these later responses tie to active functional connectivity. So if we were to activate a region, whether that should have a similar effect, I'm not sure about that particular part, but I think that these later responses definitely give insight into functional connectivity, we don't know the structural pathways underlying those yet. Only these N1 responses have been correlated with diffusion imaging by several groups. So thank you so much. Thank you so much for all the questions. If there's any other questions, this has been a great, thank you so much for being here, and thank you so much for having us today. This has been a wonderful session with many questions. I posted the links in the chat window, such that people can, if there's any follow-up questions, they can comment on the GitHub page or download the data from OpenNeuro. And
I'm told that the tutorial videos will be available on the NIMH website at some point in the future. I'm not sure how long, but you'll be able to review this if you need to.
Thank you so much. Thank you, Janice.